So as promised, today we finally have our clicker questions. So we'll get started with those right away. Clicker question 1. How many different axes of symmetry does an icosahedral virion have? None, 1, 2, 3, or 4. Love these kinds of questions. Makes it really easy for me to come up with answers. I'm sure you can stare at my soccer ball too here, if that helps. And feel free to chat amongst yourselves, by the way, in this. This is supposed to not be your own individual thing. Ten seconds if you haven't voted yet. Okay, let's look at our results. Yay, I'm glad to agree that everybody seems to agree with me. D is good. I like D. I like D a lot. And then we can go through and click through here. Yes, it's which ones? We've got the 5-fold, 3-fold, and 2-fold. Exactly. So 3 different axes of symmetry. So let's close that again. Next one. The simplest of icosahedrally symmetric virions have blank copies of the capsid protein in the virion. 1, 12, 20, 60, or 180. Again, feel free to discuss it amongst yourselves. Unfortunately, you can't do that on the exam, so. What? Yeah. Dr. Singer. Dr. Singer. Ten. Uh-oh, does he pick D every time? Maybe. Who knows? So what's the simplest of the icosahedral symmetric viruses, or I should say virions? K equals 1, exactly. And so those have how many subunits per icosahedral face? 3, because there are 20 faces on an icosahedron. So 3 times 20 is 60. Yay! So, yes. And I did hear the comment, actually, by the way, about, Dr. Singer lets you, chat with each other during exams. I have actually thought about doing just clicker questions for a whole class and doing away with exams entirely. That would be a different way to do things. Yes. So we shall see. It's just one of the things that I've been thinking about as far as this class is concerned. Okay, where do we go? Hide results. So our last clicker question for today is not icosahedral symmetry. What amino acids are most often present on the surface of virus genome binding proteins? Lysine and arginine, glutamate and aspartate, glutamine and asparagine, cysteine, methionine, valine, leucine, and isoleucine. And yes, that should be a comma. And you lose 0 points for guessing. Okay, so we've got a little bit more differences of opinion here. So what amino acids are understood on the surface of, when we have genome binding proteins? What do genomes have? Always are going to have a negative charge. So what kind of interactions do you want to have with that? Positive charges. Which of these amino acids have positive charges? Lysines and arginines, yes. So when I mentioned, and this is actually a nice sort of throwback to the first lecture, when I mentioned I expect you to know things from molecular biology, cell biology, etc., those are some of those things. So, okay. Any questions, comments, worries? No, he doesn't always do D. So at least we've got that taken care of now. Okay, that's it for clicker questions. So people need to take off. Please be quiet and run away. Otherwise, you can just hide your clickers for the time being. So sort of reviewed virus structure already, particularly icosahedral symmetry, through those clicker questions. We talked a little bit about methods last time, and really critical in terms of those methods, really having to do with electron microscopy and really the single particle reconstruction processes, because that's really how we're getting most of our reasonable resolution virus structures like this one over here, one of my favorites. We'll talk more about towards the end of today. Icosahedral symmetry, hopefully we've beaten over the head now. T numbers, we'll cover that again in just a second, so I won't talk too much about that. We looked at some of the examples last time. Particularly important are some of the exceptions, like SC40, where you've got, instead of hexamers, so 6 copies of your capsid protein, you actually only have 5, which is really bizarre, and people didn't expect it until they actually had some high-resolution structures to look at. We kind of skipped over envelope and assembly towards the end. The main reason for that is we actually understand a lot less about envelopes and that lipid coating which comes on the outside of the enveloped viruses. And then assembly, it's a very specific viral process, but it's very specific to the individual viruses themselves. And so we just sort of, a really quick overview, but again, we'll get back and talk about a lot of the differences there later on. And today, we'll spend considerably more time talking about virus taxonomy. I promised to post this test question last time that we had at the end of a lecture. T number of the virion on the right. Again, key for all of these things is finding the 5-fold axes of symmetry, which are the beginning and the end of the line here, and then wherever your hexamers are in between that. So your pentamers, 5 copies of your capsid protein, and then the 6mers in between that. So here, one direction, so your H is 1, change direction, go 2, and then you get down to here. So that's going to be a T number of 7 in this particular case. Is this clear to people? Hopefully, yes. Happy? I know a number of you came up to me after class last time and said, is that a T equals 7? Yes, it's a T equals 7. I am unlikely to give something like this on an exam. I would probably give more of an outline, as you saw last time we had the lecture, where you've got more of the actual icosahedral structure. You can see it a lot more easily in that case. This is really what they look like, but picking out T numbers is a lot easier on the other side. In terms of taxonomy, this, sorry, is a slide that I didn't post because I noticed going through this morning. I was like, oops, I didn't put an outline in here. We're not going to talk very much about virus taxonomy. We're going to spend a lot more time on virus entry today. So virus taxonomy, what most virologists think about in terms of virus taxonomy is this. The 9th report, actually they're working on the 10th report now, of the International Committee on Taxonomy of Viruses. This is actually a group founded by international treaty, and this is the official taxonomy of all viruses. And I forget how many pages this version is. Let's see, 1,327. And the next version is probably likely to be, every time they come out with one, which every couple of years is usually about 10 to 20% thicker. So it's getting a little out of control. Also, I think 90% of what is in here is garbage. So, and we'll talk about why that is in just a second. I think it's much more interesting to talk about the taxonomy of viruses, or more importantly, the phylogeny of viruses based on their genomes, rather than what's used in here that we'll talk about a little bit more in just a second. Then also, I think probably the way to move forward in terms of a real, not so much taxonomy, but a phylogeny. People get the difference between taxonomy and phylogeny? Yes, no? Taxonomy is naming, and phylogeny is relationship. So naming is really useful, but if there's no actual relationship between them in terms of their inheritance, I'm not sure how useful it really is. And so that's why we'll spend some time talking about some of those genomes, and again, why I don't like that much. And I think really the best way to do taxonomy and probably also phylogeny for viruses is to use structure rather than sequence. And that's actually really hard to do. And I'll give you a nice example of that, again, based on the STIV virus a little bit later on. So what is in this book? You know, how can you spend 1,300 plus pages on things? They also, unfortunately, not terribly well updated, but silly to print 1,300 pages these days. It's much better to have it online. So that's there. But the main criteria that are used by the International Committee on Taxonomy of Viruses is all the colored stripes you might be able to see on the back of here. But basically, what that is it says it's the nature of the genome that actually gets packaged inside the virion, which is the first criteria. And that's what you'll see all of these sections here. There'll be single-stranded DNA, or this big one right here, positive single-stranded RNA, for instance. So what kind of genome it has, the symmetry of the capsid, is it icosahedral, is it helical, is it something else, whether or not it's an envelope, and how big the capsid is. So those are the really main criteria that are used by the ISCTV in terms of classifying viruses. And you notice one big thing is missing in that list, and that's genomes. So if you really care about common ancestors of viruses, these things probably aren't evolving terribly much. And so if you care about the phylogeny, genomes is a much better way of looking at these things. And so the idea of having one of these groups is that they'll have common ancestors. And I think just by looking at these natures of genomes, it's not going to tell you anything, whereas the actual genome sequence itself will. There was a question here?
I was going to ask what you meant by, it says nature of genome, but what do you mean when you say?
Oh, nature of genome. So it's what the categories are in here. Double-stranded DNA. It'll be single-stranded DNA, double-stranded RNA. Positive strand, single-stranded RNA.
So do you mean like the actual sequence? Are you talking about what should be in there?
Right. So I think a lot more should be based on the genome sequence, and as we'll get back to a little bit later on, potentially the structure of the capsid as opposed to the dimensions of the capsid and the symmetry of the capsid. And part of that has to do with the techniques we now have to do this. And when this originally was brought up, it was a lot more different. Yeah?
That kind of answered my question. I was just going to ask why they don't use genomes.
Yeah, well, there are a couple of reasons why not to use genomes. One, actually, is that if you think about it, I showed you, I think it was in the first lecture, the universal tree of cellular life, which has the ribosomal RNA sequences in it. Well, there's no universally conserved sequence in viruses. So that's clearly one big problem. And the second one is that viruses, or I should say virus genomes, have a tendency to evolve really rapidly. And so it's hard to do a lot of these things. Yeah, your question?
I was going to ask if you could have something that was genetically similar but had different genome nature, like to this.
Okay, so I wasn't planning on talking about that, but now you mention it, I will. So there are a couple of cases, and we'll talk a little bit about this when we talk about the retroviruses, because retroviruses, you can package single-stranded RNA, you can package single-stranded DNA, you can package double-stranded DNA, and they're all retroviruses. So, wait a minute. Okay, that kind of gets away from this whole nature of the genome thing here. So, to some extent, it's always going to be a compromise. Where biology, there are always exceptions, and when you're talking about virology, there are usually even more exceptions. Okay, so again, the big question here, really, I think, if you're going to have a useful taxonomy, is going to be in terms of trying to understand the phylogeny. Taxonomy, however, is really pretty good if you're mostly concerned about things from a medical virology point of view. It does actually really work out reasonably well if you're looking at the very sort of tips of your tree. And so in that case, it does actually work reasonably well. The whole idea of a virus species, if you want to read almost as much commentary as there is in this textbook about why you should or shouldn't have virus species, that's basically in here. What's a species of a virus? Species all comes from the Linnaean concept of being able to reproduce, etc. So I don't like virus species, but something that people use. And then most people kind of agree on this. Virus species. As soon as you get beyond that, genus and family, that becomes really kind of a god-awful mess. And then beyond that, as far as I'm concerned, forget it. So let's just really briefly talk about some of these nature of genomes, and then we'll move on from there. DNA viruses, this actually, let's see, which segment is that in here? That's this big green one, the top, double-stranded DNA viruses. There are two flavors of DNA viruses. You've got single-stranded DNA and double-stranded DNA viruses. We'll spend a while talking about single-stranded DNA viruses later on because they're some of my favorite viruses because we discovered some in these acidic hot lakes. They're totally cool and really bizarre in terms of thinking about what kinds of things can be packaged. Single-stranded DNA viruses can be really, really small in terms of their genomes, and the 14 kb that I have in here is some new stuff, newer than is actually published in our textbook. Turns out there's some archaeal DNA viruses, which are single-stranded DNA. The structures of these, particularly the single-stranded DNA viruses, but the structures of their genomes are actually really interesting. And again, we'll talk about that later on. Turns out they're really everywhere, and potentially a real dominant class of viruses in the environment. A lot of those little dots that we saw before, and I mentioned, my very first lecture that some of those dots actually underestimate the number of virus particles which are out there, or virions, I should say, because single-stranded DNA actually doesn't bind to a lot of these nucleic acid binding dyes very well. So there are lots of single-stranded DNA viruses out there, and again, we'll spend some more time talking about it. Certainly in terms of the world and those little spots that you see in environmental samples, they're mostly double-stranded DNA viruses because most of these are viruses that are infecting bacteria, also known as bacteriophages, we've talked about before. These guys can have ginormous genomes, and in fact, the largest genome of any virus to date is that of the Pandora virus, which is almost 2.5 million base pairs in size, which is really kind of amazing because lots of bacterial genomes are considerably smaller than this, actually. Our favorite microbial genome is the sulfolobus genome, which is 3 million base pairs. And that's a cell, as opposed to one of these virus genomes. So what's in some of these virus genomes is a really interesting question. A lot of people talk about virus dark matter. Again, I'm not sure how many of you have got turned on to TWIV yet, but they spend a lot of time talking about viral dark matter, and we'll talk more about dark matter a little bit later on. Again, a lot of this is really broad brush because we'll be talking about the individual virus families as we move on here. RNA viruses, there's a really large section in here on the RNA viruses. Partly that's because a lot of RNA viruses infect humans and plants, which lots of people study. So there are lots and lots of described viruses in this class, way fewer of those double-stranded DNA viruses, but who cares about viruses that infect bacteria? Yawn. So the positive strand RNA viruses can be really huge, up to 31,000 base pairs, which is really interesting, and we'll talk about this, particularly when we talk about the coronaviruses, because RNA in and of itself is pretty unstable, and anytime you've got 31,000 bases that you're trying to copy, you're going to end up making a lot of mistakes when you copy these, and it turns out that there's some really interesting mechanisms on how to deal with that. Generally, they're much smaller, but again, you've got a whole different kinds of possibilities. Curiously enough, most of these have what's called unimolecular genomes. So when I talked about nature of the genome before, I talked about single-stranded DNA, double-stranded DNA, negative and positive strand. The other aspect that they use for taxonomic purposes is segmented or non-segmented. So I just like to think of segmented as like having different chromosomes, like we have. So our genome is segmented into lots of different chromosomes. And the same thing is true for a number of viruses, but particularly, curiously enough, the negative strand RNA viruses. Why it is that these guys are much more segmented, the positive strands much, much, much less so, is not entirely clear. And may have to do with replication, and we'll talk about that when we get to it a little bit later on. So positive strand, negative strand, of course, that gets back to whether these guys can code directly for protein or not, as the case may be. So negative strand RNA viruses are infamous because they cause us lots of nasty diseases. So the Ebolas, the influenzas, etc. A lot of these negative strand RNA viruses. There are a number of double-stranded RNA viruses that are out there as well. And again, another little segment here in virus taxonomy. They basically behave like negative strand RNA viruses. You could almost consider these two to be similar. Again, they've got a lot of segmented genomes, usually icosahedral capsids as opposed to helical capsids, but these are pretty gross generalizations here. So the last class that I wanted to talk about here, this is actually getting back to your differences in terms of how these things get packaged. These are the reverse transcriptase viruses, also known as retroviruses in many cases. And that's just because the replication of the genomes of these viruses always goes through a reverse transcriptase phase. So taking an RNA and making a DNA copy of it. But it turns out that there are lots of different things that end up getting packaged in your virion. So in the case of the classic retroviruses, things like HIV or a lot of the RNA tumor viruses, those package RNA. But the HepaDNA viruses, best known of these is hepatitis B, package this bizarre combination of partly double-stranded, partly single-stranded DNA. And it turns out that that's an intermediate of this replication process. And so it just packages one of the intermediates in that process. And the colimoviruses, we'll learn more about looking at some of these plant virus names a little bit later on, but coli stands for what? Cauliflower, and mo for mosaic. And so most of the plant viruses are actually named based on what they're infecting and then what the symptoms are of that disease. So mosaic in cauliflower. So the colimoviruses, which are almost completely double-stranded DNA, but again, they're replication intermediate. And then we think that this RDHV may have arose through reverse transcriptase. This is one of the viruses we're working on in my lab. It's not really, probably should put quotes around this or parentheses down here at the bottom. Probably had to have a reverse transcriptase in its past. So just for completeness purposes, I wanted to talk about a couple of other things. This is basically the only time we're going to be talking about them for the whole class. These are so-called defective viruses. These are viruses that require something else in order to replicate. And you can sort of think of these in some ways as viruses of viruses. So things that are going to infect something that already has a virus infection in it. The so-called satellite viruses, these all have to infect a cell that's already been infected by a different virus. Although if you think about numbers, again, particularly environmental, there are huge numbers of viruses out there. And in many cases, an organism is stably infected with viruses. So it's not terribly surprising that these satellite viruses manage to replicate. And then there are these really bizarre things out there called viroids. I like to think of these as really, really naked viruses because they have no capsid. They're just RNAs. And they just seem to be these parasitic RNAs. They depend on other viruses for their own replication. Often these small pieces of RNA don't even encode anything other than basically a little origin of replication that says, hey, replicate more of these. And we wouldn't know about them if they didn't cause disease. And so in some cases, particularly plants, some of these viroids actually do cause disease. And it turns out that plants are a really interesting system because all their cells are connected to each other. And so these RNAs can actually get around. So they don't actually have to have capsids in order to be able to get from cell to cell. Some people say that these are sort of the remnants of the RNA world. Whether they are or not, there's always going to be this argument, I'll get to in just a sec, whether this is a reductionist kind of process, it was something that was really big and it's now really small, or it started out small and then these things have developed from it later on. Yeah?
So do the satellite viruses have a favorite helper virus, or would a given one want to infect cells infected with a stem infected with another virus?
Yeah, so the question is basically, are the satellite viruses specific? Just to paraphrase your question here. And so that basically is, would a satellite virus of a particular other virus be there? Now, the best understood of these, probably best studied, are the so-called adeno-associated viruses, which are satellite viruses of adenovirus infection. And so pretty much all satellite viruses are going to have a partner, which you're always going to see them with. Hepatitis delta virus is a really nice example of that. Only infect cells that already have a hepatitis virus infection. There are also some examples in the bacterial world. There's some bacteriophage P2 and P4. P4 is dependent on P2. So yeah, generally they're very, very specific. And if we have a chance right at the end, we might talk a little bit about the so-called virophage, which have to do with the giant algal viruses. So that's basically the stuff which is in here. Now I'm going to tell you a little story about how I think virus taxonomy should work, or at least virus phylogeny. So this has to do with a virus that, what's the T number? Quick. Remember from last time? T equals 31. Not going to ask you that later on. But this is a virus that I discovered in a hot spring in Yellowstone National Park. And the structure was determined by Jack Johnson's lab, particularly Yang Tang, who was a postdoc in the lab at the time. They loved this virus particle. It's got nice icosahedral symmetry. You can feed it into your cryo-EM structures, and it's really easy to assemble something because it's got this nice symmetry to it. So they did that, but one of the things that they noticed, which was they ignored these really pretty projections. No, it's not a color EM. This is artificially colored. But if you look at actually what's between these five-fold axes of symmetry, they noticed something that was really, really interesting. And that was that, this is now the STIV here in the middle. You can see sort of three projections. So it'll kind of look like three fingers sticking out from each of these hexagonal subunits that are fitting together in this icosahedral face. What they noticed is that those actually looked really similar to some of the hexagonal faces in some bacterial viruses. This particular one is PRD1. It's a bacteriophage that infects Salmonella. And probably more excitingly, at least for all of us eukaryocentric types, is adenovirus. And we looked at adenovirus a little bit before. Remember the hexons and pentons we talked about before? So it turns out that the hexons in adenovirus also have this sort of tripartite structure that kind of sticks out in each of these hexagonal units. And so they continued to work on this and looked at the structures, the crystal structures of the major capsid protein that's making these kind of hexon-like structures. And what they noticed in the first time we published our paper was that this bacterial virus protein was extremely similar in its structure to the adenovirus protein if you ignored the projections up there at the top of this adenovirus structure. And so it turns out that there were these beta barrel jelly rolls that we talked about last time. We talked about a lot of the capsid proteins that come together. Beta barrel, in this case, 2 of these that came together, 2 that come together in adenovirus. And what's really interesting is you can take this, I don't know if can you guys see this in the back? There's sort of a bluish and then some colors that are inside it. Yeah, so this now is the blue, which is our archaeal virus. And these different colored pieces, all different colors up at the top here, and then slightly similar orange and reddish down here. These are actually now the structures from a bacterial virus put into this archaeal virus structure. And what's really amazing is they fit perfectly, which is really bizarre because the sequence now of these things is not similar at all. So how can these things be related to each other? Well, the structures are basically identical. So how could this actually be happening? Certainly it's a possibility. It would be horizontal gene transfer. Hopefully you've heard about horizontal gene transfer either in principles or in microbiology. This is where stuff gets moved around from various different hosts. What do viruses do? They move nucleic acid around, so it's certainly a possibility. But you'd have to have horizontal gene transfer between archaea, bacteria, and eukaryotes. Seems rather unlikely. Another possibility is that you'd have convergent evolution. And this is actually kind of getting back to your question about different kinds of things being packaged and how do you do this. Certainly a possibility. We could have convergent evolution just giving you a particular structure, but turns out there are lots of different ways to do this. And so we think this is evidence for a common ancestor of these different viruses. Now, there's no genome sequence which is similar. Turns out that all 3 of these are double-stranded DNA viruses, but that's about it. Yeah, you have a question? No. Okay. So we published this, a few years ago, and since then we now actually have an X-ray crystal structure of the major capsid protein from our virus, and it looks, basically identical. Oh, and that's that crystal structure paper. So we think, sorry, I must have missed a slide here, so I'm back up here, that this is evidence for there having been a common ancestor to all of these different viruses down here back at the origins of the 3 different domains, probably billions of years ago. But the real question is going to be, how are we going to try and get phylogeny and or taxonomy out of structure? So that is, I think, the direction that I think we need to go. It's just a question of how we're actually going to get there. So to review our virus taxonomy, ICTV, okay, it's fine for taxonomy, thinking about usually, again, individual virus diseases, lots of generalizations, talked a little bit about these different viruses, and then what I think are some of the really important and new methods in terms of understanding virus taxonomy. Okay, so let's move on, talk a little bit about virus entry. We'll continue with this, of course, on Wednesday. We'll see where we get to. So I had my graduate student, Jim Laidler. Anybody taking microbial ecology this term? Some of you? Yeah, you've got Dr. Laidler teaching that class. So he gave a couple of guest lectures this class, and he did the virus entry one a couple of years ago. And And he calls it the heist and the challenge, which I just liked so much to not actually, switch back to my old stuff. So the whole idea with the heist, of course, is that viruses are obligate intracellular replicators. So they somehow have to find the right cell, get into that cell, and avoid the defenses that are present inside that cell. At the same time, they have to do this with only the energy which is stored in the structural proteins or the nucleic acid inside the virion. And so this is tough. This is really a hard thing to do. And then there's all this business about how the viruses replicate, but that we're going to cover a lot later on. So what about virus entry? Really mostly has to do with receptors. And so the whole concept of virus receptors, that's a cellular macromolecule that's on the outside of the cell. That's what the virus interacts with. And it's that interaction which leads to the virus being able to release its genome into the cell and go through that second part, the intracellular part of a virus life cycle. So we'll talk about membrane fusion. It turns out, again, we've got these two very overall general classes of viruses, enveloped and non-enveloped. Well, if you think about cells, they're almost all enveloped. They've got a lipid membrane around the outside. And so if you have a lipid membrane of your virus, a lipid membrane of the host, likes like, and so it's actually relatively easy to get that fusion to take place. And we'll look at some really well understood processes on how that happens. Sometimes, if you're an enveloped virus, that's going to lead to just releasing the genome if you just have an envelope around the outside. But as we talked about before, most viruses don't just have an envelope. They've also got a nucleic capsid on the inside. So you also have to then open up that nucleic capsid, also known as uncoating. In many cases, you actually have to transport that genome, or sometimes even the virion or the nucleic capsid, to a different part of the cell. That's usually the nucleus if you're talking about a eukaryotic cell. If we get a chance, we'll talk about some antiviral drugs and some defense mechanisms, but that's probably going to be on Wednesday at this rate. So what do you need to do? Now, get through the membrane. That's what cells have to protect themselves from the rest of the world. That can either be the cellular membrane on the outside of the cell, your plasma membrane, or in many cases also a vesicle membrane. And we'll see what that means in just a second. How does this happen? Almost always through so-called viral fusion proteins. And we'll talk about fusion proteins for lots of different viruses we move through. We actually know quite a lot about these at a structural level. What happens with these, again, so-called fusion proteins, is that there are these big conformational changes that happen in these fusion proteins, allowing the membrane fusion to take place. And again, we know a lot about these. We'll take a look at them in just a second. The other process, of course, is non-enveloped viruses. Somehow, you've got to get through a membrane. And there are a number of different ways of doing this. Some is literally breaking open that membrane. Of course, this is a problem if you are coming through the plasma membrane, because if you break open the membrane, you lose all kind of proton motive force. The cell dies before you even have a chance to make more of yourself, more of your virions. Again, in a totally anthropomorphic sense, but you can see that if you break open the membrane, it's going to be a problem. So usually, when these non-enveloped viruses come inside the cells, they break through the membrane once they're actually inside the cell, once they've passed the plasma membrane. And so that process of passing the plasma membrane usually happens through endosomes. The other possibility, and this is what almost all bacteriophage do, is literally make a hole and poke their genome through it. So those are the ways that you can actually get in. First problem, of course, you need to find the right cell. And so this is also what's known as virus tropism. Turns out that there are lots and lots of cells that are out there, particularly if you think about some kind of human virus. Only some cells are going to have the right kind of extracellular macromolecule, so you can bind to that cell, and B, actually have the right kind of molecular machinery inside the cell to be able to replicate your virus. So this is what's called tropism. It's finding the right cell. And finding the right cell really has to do with the right kind of macromolecule on the surface, the virus receptor protein. And this is an important point. I don't want to belabor it too much, but these are all normal cell surface molecules. It's not as if the cell evolved something that says, hi, I am the virus receptor. Come and bind to me because I want to be infected. So although if you think about it from sort of the point of view of viruses might actually be positive for an evolutionary point of view, maybe actually there is some positive selection for that. But at least most of the viruses we look at, discovered because of disease formation, they're not going to be having something on the outside of the cell which is evolved to interact with that virus. The virus is evolved to bind to a normal cell surface kind of macromolecule. So they're all normal cell surface kinds of macromolecules, and basically pretty much anything you can think of on the cell surface is a virus receptor. We've got proteoglycans, glycoproteins, glycolipids, virus proteins, adhesive proteins. Basically anything that's on the outside of a cell can and has been used by various different viruses as virus receptors. And I want you to notice I skipped the first one on here, immunoglobulins. Turns out that many of these extracellular molecules on the outside of cells have a particular protein fold, a domain-like structure, which is really similar to immunoglobulins. And the reason for that is, well, what are most of these extracellular things important for? They're important for binding to hormones, binding to some kind of extracellular factors, binding to matrix proteins. And so binding is something that most of these extracellular molecules are doing. So viruses really take advantage of that. And it turns out that the immunoglobulin fold is a really good one for binding to things. So if we look at virus receptors, we'll find that many of them have these immunoglobulin-like folds that are associated with them. And that's actually shown here. This is from our textbook. Each of these little orange pieces here does actually represent one of these immunoglobulin-like folds. So you'll notice that many of them do. This is for HIV, CD4, the poliovirus receptor, Coxsackie and adenovirus receptor. We'll talk about these a lot more later on. Many of these have immunoglobulin-like folds, but not all of them by any stretch of the imagination. So there's some peptidases, really curious, the coronaviruses, the really big single-stranded RNA viruses, the largest ones that are out there, also infamous for SARS and now the MERS-CoV. Have you heard about the Middle East respiratory syndrome virus? Yeah, so also one of these. All bind to these peptidases on the outside of the cell, which is really bizarre. Why would you bind to something which is going to chop up this virus receptor protein? We'll see why that is a little bit later on. So basically, the take-home message here is there are lots of different ones, but many of them have these immunoglobulin-like domains that are associated with them. Let's look now at a very much more specific interaction between these two, probably the best understood of the receptor binding protein interactions with the receptor. When I say receptor binding protein, that's going to be the viral protein, and the receptor is going to be what's on the cell surface. They interact. This is now one of the monomers of the trimeric influenza hemagglutinin protein. Again, probably the best studied of any of these receptor binding proteins, how it interacts with this receptor. Interestingly enough, influenza hemagglutinin doesn't interact with the protein at all. It just interacts with oligosaccharides that are present on a lot of the proteins. So many proteins on the outside of the cell and many viral proteins are modified by glycosylation on the outside of the cell. And so this particular case, you can see right up at the top there, each of those orange bits with the yellow end on them, those are the modifications, this glycosylation process, which takes place on many, many, many external proteins, et cetera, on the outside of the cell. So a lot of these, even cellular receptors, are also going to be glycosylated. And sometimes it's going to interact with that glycosyl group, the receptor binding protein, and sometimes it'll interact with a protein much more specifically. These can interact either with things that are sticking out or things that are sticking in, and we'll look at a number of the cases of that. Most of the time, once you have an interaction of a receptor from a virus, you're going to have receptor-mediated endocytosis. This is, again, it's a very cellular process that takes place. And hopefully, again, getting back to things I expect you to know, if you haven't taken cell biology, one thing to go back and look at is receptor-mediated endocytosis. So, because it's a very common process in terms of viruses. Let's look at some examples of this. Here are a number of Ig domain or immunoglobulin-like domain proteins. Up there at the top on the right here, this green one. These are two of these immunoglobulin domains which have these loops at the end. Those loops in immunoglobulins are really interacting with your antigen, so antibody-antigen interactions, usually present on the really tips. And think about Y structures for immunoglobulins on the tip. So this is an interaction point that you have here. If you look at the interaction of one of these immunoglobulin-like domain proteins, this is a cellular receptor, ICAM1, binds to lots of different viruses. You see at the end here, there's HRV, etc. That interaction, as it turns out, is right in the middle of the icosahedral subunit of one of these viruses. Here, just for orientation purposes, you can see the 5-fold axis of symmetry up here. Here's a 3-fold axis of symmetry. There's a 3-fold axis of symmetry. 2-fold in the back here. This receptor with these loops right here is interacting right there. Curiously enough, there's also the poliovirus receptor, which has this normal same kind of structure, and the loops at the end here. Well, it turns out that the poliovirus actually interacts on the side of this. So it's not just the ends of these immunoglobulin-like domains like you have up here at the top. You also see some on the side, and that's actually this MAC1 domain right here, which interacts on the side of some of these things. So immunoglobulin domains, they're really good at binding things. Our adaptive immune system seems to have taken over them in order to get antigen recognition, but viruses are taking advantage of the same thing. They're nice and sticky. Again, they're all cellular processes, particularly endocytosis, but once you get in, vesicle trafficking, nuclear transport are also exactly the same kind of thing. So let's look at getting across the plasma membrane. If you're really small, and this would be for just cells have to pick up nutrients from the environment. They need to pick up any kind of signaling molecules from the environment. They do that either by really large phagocytosis processes. This particularly happens with immune cells. Or really small molecules. This is often going to be your solutes. It's usually too small for viruses, although we'll see some cases here. Much more common are these receptor-mediated endocytosis. So you have a ligand, that receptor protein. Once you have binding to the particular ligand, then you'll have incorporation of whatever's inside there inside the cell. So these are processes that the viruses, again, take advantage of. This slide is basically everything you need to know about virus entry. So in terms of thinking about exams, etc., later on, we'll go through every single one of these later on, each of the different viruses. But basically, there are a couple of important things to see on this slide. Mostly that almost all of these are coming into the nucleus to replicate. So they have to get their genomes to the nucleus to replicate. There are lots of different ways that they can actually get in. One is in the case of HIV, there are actually specific importing proteins that get them there. Influenza virus has a matrix protein that's important for getting things in the nucleus. In some cases, like adenovirus, there's actually specific transport. It uses the microtubule system to move the virus into the nucleus. The same thing is true of herpes viruses. Hepatitis viruses seem to diffuse and end up in the nucleus. This hepatitis B virus in particular is relatively small. The whole capsid can actually get inside the nucleus. Turns out that most capsids are actually a little bit too big for that. A lot of times you actually have disassembly of your virus particle at the nuclear membrane. That's true for adenovirus and herpes viruses. So, basically a lot of these guys need to get to the nucleus. Why they need to get to the nucleus, we'll get to in just a second. There are some exceptions to this, and it turns out that the picoRNA viruses, some people also call these picornaviruses. No idea why that is. I think it's because they want to make themselves sound more important. But picoRNA viruses, small RNA viruses, most of them actually act a lot like bacteriophages. They release their genomes right at the outside of the cell. Most of these picoRNA viruses are non-enveloped, and so they end up coming in through this non-fusion process. How does that happen? Again, almost always for these naked viruses, it's an endosomal uptake. Sometimes, again, you'll just make a pore on the outside, like the bacteriophage. You've got to get in somehow. Again, you've got to get through these cell membranes. Curiously enough, a lot of viruses actually express proteins which cause membrane fusion. It's not terribly surprising, but this has actually been probably one, I think, of the best examples of how a virus protein has actually been co-opted for cellular evolution. Fusion of adjacent cells is great for a virus because it makes it easy to get from one cell to the other, but turns out that syncytium is also really critical for development of things like the placenta. So mammalian cells, certainly mammals that have live birth, are dependent on formation of syncytia, which turns out comes from a virus protein originally. So the syncytium, which is used to develop the placental, comes from this same thing. So getting from cell to cell is great for a virus, but it's also great for transferring nutrient from the mother to the developing fetus. So exactly the same thing there. Plasmodesmata are great because plant cells are all connected to each other. So it's really easy to get from one cell to another. In terms of a lot of plant viruses, they encode these so-called movement proteins. And so they're literally proteins that get moved to plasmodesmata to help move the particles through there. So that's all we're going to talk about for naked plasmid entry for now. Spend the rest of the lecture today talking about enveloped virus entry because there's some totally cool videos that I'm not sure if you guys have had a chance to look at yet. So enveloped virus entry, two membranes, one around the virus, one around the cell. You have to get these two guys to fuse with each other. And so that process, again, is almost all due to the presence of fusion proteins which have a conformational change. Now, how do you get a conformational change to take place? Most cases, this is due to pH. If you think about receptor-mediated endocytosis, one of the things that happens once you have an endosome, that lowers the pH, and that lowering of pH is probably important for stopping the activity of a lot of pathogens and also helping break down, usually whatever it is, which is food, which has come in through these endocytosis process in order to break things down, use the precursors. Turns out that many virus fusion proteins have taken advantage of this, and it's that pH change which is sensed, leading to a structural change in your fusion protein, leading to membrane fusion. You can also have just very specific receptor interactions. So the receptor interaction with the receptor binding protein can also cause this conformational change. So now let's take a look at a couple of examples of this. First one is virus entry into animal cells. And hopefully you can click on this. It'll go to the animation, I think. Here we go. This is just from McGraw-Hill textbook, but I quite like this animation because it sort of gives you a bit of an idea on a general process on how this is actually happening. So here we have our friendly enveloped virus up at the top here with all of its green little receptor binding protein. Here's the red receptor, and then once you have those interactions with each other, you release the nucleocapsid. Then magic occurs, the nucleocapsid disappears, and now we've got our genome which gets replicated. That's one process, and again, what's happening here is you're getting fusion at the plasma membrane. You can also have a full receptor-mediated endocytosis, where now you get this enveloped virus. Now you've got two membranes. This now fuses on the inside of the cell. This has the great advantage that you're not making a hole in that plasma membrane in the first place. And so that's a big thing. And it turns out that this is the process that's also used by a lot of the naked viruses. You got up through this whole receptor-mediated endocytosis. Once it gets in here, again, in many cases, there's a change in pH. That change in pH leads to magic. And then the genome is released here. That's just shown here in, again, cartoon form, where we have receptor binding up at the top here, the ligand in blue. This could be your virus particle. Comes in through the regular receptor-mediated endocytosis pathway. This is clathrin-coated pits. That endosome is at a neutral pH. As soon as it gets into the early and then late endosomes, these are processes where that pH is going down. And it's that pH going down which, again, usually leads to activation of a lot of fusion proteins. You can actually see the receptor-mediated endocytosis in thin section electron microscopy here. This is where you have receptor binding to particles, and then that causing your break-off of one of these clathrin-coated pits. Now that's the first step. The second step is here, where you can actually get the release from the endosomes. Here you can see some virus particles. These are herpes viruses inside these endosomes, which then can get released over here, and now these individual nucleocapsids are free inside the cytoplasm of your host cell. We'll look at a cartoon of these fusion peptides. Again, up at the top here, the best understood of these, again, is influenza hemagglutinin. Interacts with, at the very top here, your cellular membrane. We'll talk about the individual HAs a little bit later on, what they all do. Once you have this interaction, then you have an insertion of your fusion peptide into the cellular membrane. If you look at the sequences of these fusion peptides, they generally have very hydrophobic amino acids. Might want to remember what the hydrophobic amino acids are. Hint, hint. And then have these long alpha helices that are often not ordered before you have the interaction of these two things interacting with each other. These are the so-called class 1 fusion proteins. So it's the conformational change, once you've inserted into the membrane, that pulls these two membranes together and eventually causes complete membrane fusion to take place. So look at this in more detail. There's a really nice lecture by Stephen Harrison. He doesn't have a Nobel Prize yet, but he probably will relatively soon. Done some really fabulous work on virus structure and really has led the understanding of how these virus fusion proteins actually work. These are relatively long. It's about 20 minutes, this particular one, and then there's also a virus structure lecture, which is here. If you're interested in more information about virus structures, that's in here. But as far as today's lecture is concerned, I wanted to look at the influenza hemagglutinin protein. This is the influenza hemagglutinin protein when it's originally produced. There's a proteolysis that takes place, which gives you two separate proteins. It cuts right here in this yellow piece right here, and it's that yellow piece right here which is going to turn into the fusion peptide piece. So let's look now to finish up at an animation of this whole process, which is here, molecularmovies.com, where we have, this is a slightly different fusion process. Let's see, here's, this one is the one I want. So this is the HIV entry process here, where we have now, this is your virus down here at the bottom. Up at the top is your host cell. So we have the very often trimeric proteins which are here. This is that one from HIV. Turns out that influenza is extremely similar. This process, here's just one of those. The actual fusion process depends on pulling basically this membrane up to the host membrane, which is going to be on the top there. The way that happens is it goes from really unstructured to forming a long alpha helix. And it's that process, the formation of this secondary structure, which really pulls the two membranes together. And then this is the process here shown with two molecules. It could be one. People argue about which one is which. So now you've got the cellular membrane up at the top here, viral membrane down at the bottom. You've had this interaction. The viruses are present there. That's the first step. We have these interactions. Now you see the conformational change, which is happening here, where you're getting these long alpha helices which are formed, pulling these two membranes together. And now they've been pulled together. This is, again, the cartoon process. It's the hemifusion intermediate. So you've just got the two membranes that have fused, but there's no hole between the two. That hole is going to allow the genomes or the nucleocapsid on the inside. Here again, the viruses at the bottom, the cells at the top. These guys then pull apart in a secondary process. Now you have these two membranes which have completely fused. Now you can get your nucleocapsid to come out through the outside. And this has all been really quite nicely shown, mostly in work from Stephen Harrison's lab. And again, he spends about 20 minutes talking about that whole process right at the beginning of that YouTube that I talk about there. The end of this is actually about some antiviral drugs that you can use, and we'll start Wednesday's lecture actually talking about those.